The chapter biomacromolecules, so structure of proteins, that is primary structure, secondary structure, tertiary structure and quaternary structures. And in the diagram you see primary structure of a portion of a hypothetical protein, that is N and C refer to the two terminal of every protein. So N is the first and C is the last end. Okay, single letter codes and 3-letter abbreviations for amino acids are also different. So you see in serine hydroxyl methyl is present, and then SH, that is for cysteine. Okay, tyrosine heterocyclic ring of alcoholic, right? Then glutamic acid, that is COOH group present. So N-2, N-1 and N+1. So this is the amino acid residue you see in the diagram. So now in secondary structure, alpha beta splitted sheet. So now cartoon showing a secondary structure and a tertiary structure of proteins in the third diagram. So N and C. Now a secondary structure you see in the first diagram. So that's the secondary structure, alpha and beta plated sheet. Now coming to this, that is how do, like, okay, so in the chemical reaction, okay, where is that? Okay, so in the enzymes like chemical reactions you see barium hydroxide plus H2SO4, that is barium hydroxide, barium sulfate plus water. So rate is equals to rate of product upon rate of time, that is CO2 plus H2O. Carbonic anhydride form carbonic acid, glucose 2-pyruvic acid, C6H12O6 plus O2, C3H4O3 plus water. Now how do enzymes bring about such high rates of chemical conversions substrate to product? Because they bring down the energy barrier. Now concept of activation energy, potential energy on y-axis, x-axis progression of reaction you see substrate to product going through transition state. Now with activation energy or without activation energy. So with activation energy it is lower. Okay, now nature of enzyme action you see enzyme plus substrate, then enzyme substrate complex, then enzyme product complex, then enzyme plus product is formed at the end. Now factors affecting enzyme activity, temperature and pH you see in the diagram. Effect of change in pH and temperature you see there is optimum. On y-axis it is enzyme activity, on x-axis with respect to pH and temperature, and then concentration of substrate on enzyme activity also affects you see on substrate. On x-axis that is Km value that defines velocity of half, and velocity maximum you see velocity of reactions. So this is it. Now